Good afternoon, everyone. Continuing the activities of the second Brazilian Symposium on Fractional Calculus, I have the honor and the pleasure to bring you directly from University of Illinois, Chicago, Professor Richard Magan, a fundamental reference in bioengineering with his famous books, Fractional Calculus in Bioengineering, and several important articles. Advisor of several masters and doctors, and with more than 15,000 citations in academic Google, Professor Richard Magan is one of the legendary scientists of fractional calculus. Professor, thank you very much for kindly accepting our invitation. We would like to be in person with you here in Brazil, presenting you some of our natural and cultural wonders. But as we cannot, we are very happy with your virtual presence here with us. Thank you once again. Nice to have you here, Professor.
Thank you very much. I'm looking forward to presenting and sharing some of my work and my colleagues' work, and also to answer any questions and interact with the audience. And what's the next step now?
Well, now we are going for your presentation, and I'll leave you.
Okay, so let me start my slides, I hope. Okay. Can you see my first slide, Ruben?
Yes, it works perfectly.
Okay, good. And so my talk today is going to be on the applications of fractional calculus in magnetic resonance imaging. And I am looking forward to sharing the work that we've been doing, and I'll try to do so in a reasonable time frame, I hope. But I'm like you say, it's too bad we can't do this in person, but I'm happy to be able to do what we can. First of all, before I start, I'd like to thank Professor Jose de la Sosa and Professor Rubens Camargo for inviting me to present this talk, and to the SBMAC for their support for this symposium. Rubens, if you or anyone else has a question, just say it, because I have no visual feedback here, and so I don't want to go off on a tangent.
Okay,
Professor. Good. Okay, a summary or synopsis of the talk. This is a cartoon I got from a British magazine. Gentlemen and gentlewomen, this is just to give you an idea. Actually, there are lots of wires and things. What I'll be talking about is largely an overview, and the basic idea is to show how fractional calculus models, particularly of diffusion and relaxation, can be used to capture the complexity of brain tissue as it's revealed in MRI. The outline of the talk is as follows. I'm going to divide it into three sections. The first section is context. I'll try to put the work we're doing with fractional calculus in MRI into its historical context. Next, I'll try to establish connections between MRI and fractional calculus. And then at the end, if we have time, I'll speculate a little bit with conjectures about what aspects of this could be continued.
Let me interrupt you just for 1 second. It would be nice for us if you could click on this hide. This is little bottom here, hide.
Ooh, I worry about hide. Yeah, I saw that button there, and I really didn't want to do it, but I did it. Now what do I do?
Thank you.
Now, wait. Okay, now we're ready to go. Okay, good. Thank you. Yeah, I saw that there, and I was tempted to hit it, but then I was figuring, well, it's annoying, but if I hit hide and everyone goes away, it's going to be very sad.
No, it was fine.
Okay, good. But thank you. Okay, so let's get on with it. Context. I'm going to use a Venn diagram. I'm going to look at the fields of fractional calculus, which is the subject of the symposium, magnetic resonance, which is the field in which I have been mostly applying fractional calculus, and then the sort of unifying or overlapping area of random processes, which is something I don't really understand, but it certainly has overlap with both fractional calculus with things like the continuous time random walk model and with magnetic resonance. So magnetic resonance, as you may or may not know from learning something in chemistry about nuclear magnetic resonance spectroscopy, and in bioengineering about magnetic resonance imaging, is used to, at one extreme, probe molecular structures, and at the other extreme, make pretty pictures of your knee, your liver, and your brain. Fractional calculus, you are all familiar with to some extent. It involves different models of generalized non-integer order operators. Most of what I do follows the work of Metzler and Klafter and Minority in using the continuous time random walk model. A little bit of fractional motion, but I'll make that clear which one of those models we're using. Random processes, it is the basis of thermodynamics. Heat, for Pete's sakes, is actually just random motion. Diffusion and relaxation in the cases of MRI involves molecular and translational motion of molecules. Principally in MRI, it's the motion of the water molecules that is most of most interest, partly because we're mostly water, and partly because the water is able to probe intra-extracellular environments in ways that provide changing signal intensity that can be used to generate contrast. But I'll tell you more about that later. Okay. Connections with fractional calculus. So I think it's good to understand the overall context of where we're going, but the connections between what you know and what I hope to discuss and perhaps what you might want to learn today is the purpose of this chart. So fractional calculus has applications almost everywhere, that there's a complexity in the material or the processes. It was pioneered with the work of Oldham and Spanner in electrochemistry. It's been applied in viscoelasticity and in dielectrics. We'll be talking today about its application in magnetic resonance. The thing about fractional calculus, as you know, is there's nothing fractional about it. It's all calculus all the time. It just allows you to generalize the kernels of integral equations in such a way that you can include non-local events in time or space. And so you're messing with the math, which gives you insight, and it also provides some complications. In MRI, we'll apply it to the particular field of imaging. Magnetic resonance is also a large field. Chemists do nuclear magnetic resonance, and a lot of physicists do what is called electron spin resonance. We'll talk about MRI, and within the MRI field, I'll be looking at relaxation and diffusion. Those of you who have done any applications with fractional calculus typically look at generalization of the models of relaxation in a viscoelastic sense, or diffusion in a molecular sense. In MRI, the relaxation follows classical models of Gaussian or normal diffusion, leading to exponential decays with characteristic time constants, T1, T2. In fractional calculus generalizations of relaxation, so-called anomalous non-Gaussian diffusion, we all know that the exponential becomes generalized to the Mittag-Leffler function, and you still get time constants, but you also get the order of the fractional operator in time that modulates these effects. And a simple way to look at this is that it introduces a distribution of relaxation times. Diffusion is a big part of clinical imaging, in particular, the imaging of orthotropic tissues, muscle, connective tissue, spinal cord, white matter in the brain. That is, you can use the restricted diffusion to map out the structure of tissues. And those models, again, they're Gaussian. You get a diffusion coefficient, which is directionally dependent. So it becomes a diffusion tensor. And you can make images of this distribution in space as diffusion tensor imaging. With fractional calculus, we allow the diffusion to be non-Gaussian. We generalize the diffusion equation to involving space and time fractional derivatives. In the non-Gaussian case, we get to choose those derivatives in the fractional world. I'll use in my talk what I've done mostly in my application, which is involved the Caputo derivative for time and the Riesz derivative for space. So you get the diffusion, you always get what you start with in relaxation or diffusion, but you get extra parameters that in the case, the alpha and the beta here, I will relate or connect to fractional time and space derivatives. You still with me, Ruben? Are you still there?
Yes,
Hello?
Professor.
Okay, good. I just, okay.
Just fine.
Okay, good. Okay, so first of all, I'll talk about relaxation, and I'll be looking in general at the intersection of all three of these fields: fractional calculus, magnetic resonance, and random processes. I'll look at relaxation first, and then I'll look at diffusion for the rest of the talk. I guess it should be obvious, but I'll just list the names, that the work we're talking about here is basically a who's who of mathematicians, physicists, and engineers. If you look within the field of MRI, Bloch Purcell, who discovered the NMR phenomena, and then it was later extended into magnetic resonance imaging, those pioneers provided the foundation of this molecular probe. Fractional calculus, Riemann-Liouville, Grunwald-Letnikov, Oldham-Spanner, Mittag-Leffler, Caputo, I can go on. You guys know all these. Random processes, it's hard to underestimate the significance of Einstein, Smoluchowski, Fokker-Planck, Langevin, Markov, Gauss, Poisson, Wiener, and Levy. All of these mathematicians contributed to extensions of physical concepts in such a way that one could get a better understanding of processes as they are extended in time and space throughout all of physics. And now, I will show in some of my work, as it applies in bioengineering and biomedical imaging. The Bloch equation is the fundamental ordinary differential equation that governs magnetic resonance phenomena. It's a simple, well, it's nonlinear actually, but in a linearized form, it's a simple precessional motion of a magnetic field, M, in the presence of an applied field, a B, in such a way that the orientation of the vector can be manipulated and localized. Anything you do to the magnetic field means you're operating on this vector cross product here, involves all sorts of components that can either cancel or reinforce each other. But no matter what you do, when you stop doing it, or even while you're doing it, there are inexorable relaxation processes occurring. And these T1s and T2s basically are just reflecting the first and second law of thermodynamics, that the system, if you leave it alone, and even if you don't leave it alone, it will tend to equilibrium, and it will tend to the lowest energy configuration, and it will tend to maximize entropy. The precessional motion of magnetic fields involve a concatenation of static, time-varying, linear, and linear variations in magnetic fields. The net result is that when you apply a radio frequency pulse at a proper frequency to a magnetic the active nuclei, the M0 vector here, which here is oriented along the y-axis, can be tipped to any orientation perpendicular to the direction of the magnetic field. As soon as you've done that, the magnetization just spirals back around and around and around until it converges back along the z-axis. And this rate at which this happens is the so-called Larmor frequency. In a conventional MRI of, say, 1.5 Tesla, this is 64 megahertz. It's like channel 2 or 3 on US TV. It's RF. And this process of relaxation decays in time following T1 and T2, which relates to either the Z or the XY component. I teach a whole course in this. There are books in it. We're going to, of course, do the usual trick. We're going to mess with the math. We're not going to try to mess with the fields. We're going to try to change the order of this time derivative. And so what we're going to do is we're going to replace the time derivative by a fractional order time derivative. And I'm assuming that in other presentations and in other coursework or in other readings, you have encountered different kinds of time derivatives. I'll focus principally on the Caputo time derivative. And for this particular process, since there's a T1 and T2, I'm going to specify that the T1 and T2 could have a different order. That is, the T2 decay might follow an alpha time derivative, and the T1 a beta time derivative. All through fractional calculus, time and space and different forms of derivatives are typically represented by an alpha or beta. You got to be really careful, because here I'm talking about alpha and beta as relating to the T1 and T2 relaxation time. This is not always the case. You have to always be careful to make sure that what you think the terms relate to are in fact what the author means. Speaking of the author, this work was written up, I guess, a year or two ago in a review article in the Critical Reviews of Biomedical Imaging. And for details, you can go and read there, or you can go back and listen to some of the talk or read some of my other papers. Of course, what happens when we apply a fractional model to a relaxation process is we convert the exponentials into Mittag-Leffler functions. Here, I've just assumed that we're going to leave the Z component alone. We'll just use a fractional derivative alpha here on the XY components. And so what happens is you still get this helical motion after you perturb magnetization, in that it returns to its orientation along the Z axis here. But the rate at which it returns, that is the decay rate, follows the Mittag-Leffler. And the Mittag-Leffler, of course, falls faster than an exponential at early times, and then falls like a power law, T over T2 star to the alpha for longer times. So what one finds is that in addition to the T2 characterization of the process in the exponential sense, you have a decay rate that also varies as the alpha. So as the alpha drops from 1 to 0.9 to 0.7, 0.8 and 0.7, you get a tighter or faster decay. This model has been applied to look particularly at complex tissues, where the usual exponential model doesn't work. So the next step is almost often a multi-exponential model, but exponentials more or less always go away eventually. The fractional order models with these power laws can persist for quite a long time. Okay, so since relaxation in time is fairly well understood, I thought I would, for the rest of the talk, talk about the use of fractional calculus in diffusion, where both space and time derivatives play a part. How are you doing on time, Ruben? You still there? Yes? No? Ruben, am I still here?
Yes, you're still here.
Okay,
and
I was
still
scared.
having time.
Oh, okay,
My microphone was closed.
Okay, no, sorry, I was getting really worried. Okay, good. Okay, well, so I've talked about relaxation. Now I'm going to talk a little more about diffusion.
Okay.
The diffusion issue is interesting because there's basically a mismatch. When you look at an MRI or a picture, a virtual slice of the brain of a real brain, in this case an excised fixed brain from an animal, when you look at an MRI, you see these slices. The field of view is typically the full size of the head. The pixel resolution is on the order of millimeters. But when you look in a biology or a pathology book, when you look at the structure of tissues, you see cells. Cells are on the order of a micron. So when you look at microscopy and you ask questions about cellular tissue organization at the cell level, you're at microns. So it's a factor of 1,000 at least, maybe 10,000. So this is the place where fractional calculus comes to the front, because we take the low resolution MRI, the high resolution structural model of tissue, we use fractional calculus to inform a geometric model of the structure. In this case, these are bundles of axons in the corpus callosum that connects the right and left halves of the hemisphere together here. You can use fractional calculus to account for things like permeability or distributions of diameters within this connection. And then this fractional calculus model is fit to not just one image, but many, many images collected by varying the parameters, not only in direction, but in terms of diffusion time and diffusion sensitivity. The net result of the fitting process is that you still get the picture that you started with, except you get colored dots. And the colored dots are basically the values of the fractional parameters that you have fit to the model. So where you see red dots here or yellow dots here, you have higher or lower values of your fractional order, say alpha or beta, in your model. And so the idea is that the fractional model allows you to describe complex structures that you use as a tool for describing the microstructural subvoxel organization of what you see in the image. And then the $64,000 question is, as disease or age or other development occurs, it's the microstructure that changes. You can't see that necessarily in the conventional MRI, but you can see it if your model is sensitive to those parameters. Okay, so how does this work in MRI? Well, you start with a block equation. Everything begins there. And you add diffusion and you add flow. And so I won't talk about it, but magnetic resonance is great for imaging flow. And diffusion is imaged by appending to this time rate of change of magnetization the motion of the imaged nuclei as it diffuses or as it flows. You do this by applying more radio frequency and gradient pulses. And the idea here briefly is that you apply a pulse and you spin label particular parts of your brain or parts of your image, and then you flip the whole thing over and wait a little bit, and then you apply the same pulse again. And if the sample hasn't moved, if there's no diffusion, if there's no flow, you get nothing. There's no change. But if there's been flow, if there's been change, then you get less of a signal than you would if there had been no motion. So you get a shift in phase or a shift in amplitude. Principally, the flow gives you a shift in phase. The phase that comes about with the diffusion pulses is resolved as a change in amplitude. So if you just look at the XY component here of the magnetization, and there's a T2 relaxation term. And so now what we're getting here is we've got a Laplacian operator here. So we have a spatial variation in this magnetization. We have a T2 relaxation, we have a precessional motion, and we have time rates of change. If you fiddle with the parameters somewhat, you can actually solve this and get a simple exponential. And the exponential decays as a function of parameters you can control. Everything in this line here corresponds to a width or an amplitude of a pulse in that picture. So these are either the physical factors or the system parameters that you can adjust. And so when you adjust those parameters, you will see the signal decay, and it will decay at a decay rate, and the decay rate works out to be exactly the diffusion of the labeled molecule in the sample. So life is good. It's a simple exponential. Until you actually look at some different tissue. Here's some signal collected from the brain of a human subject, actually. This is a cerebral spinal fluid, the water that bathes the tissues in the brain and the spinal cord. The gray matter, what we're thinking with now, hopefully, it shows up in the cerebrum and cerebellum, and then the white matter is basically the connecting cables, if you will, between the functioning units within the brain. And you can see here, this is plotted on a semi-log graph. So B is the parameters that you select when you're imaging, and the slope of the decay here, since this is a semi-log plot, this is a log scale, shows up as a straight line if and only if it's an exponential. If it curves a little bit, it means that the diffusion coefficient may be changing as a function of B or time. If it curves a lot, it means that your model is not right. And what we see here is, of course, water is water, and the CSF decays linearly, and the decay rate is very close to the known diffusion coefficient of water at that temperature and under those conditions. The gray matter looks linear for the most part until you get to really high B values on a semi-log axis, but the gray matter doesn't. So this becomes an opportunity for applying fractional calculus. We could apply multi-exponential models. That's what everyone did first. We could apply a stretched exponential model, which we all know is just a crude approximation of a Mittag-Leffler function, but they give you some information. Or we could just boldly go where no man has gone before, or woman in the Star Trek sense, and try to capture this with some sort of fractional model. And that's what I did. I said, okay, the Bloch-Torrey equation, at least for the xy component here, has a time derivative, it has a precessional component, and it has a space derivative. And I said, why don't we just generalize the time derivative to a fractional time, and generalize the space to a fractional space, and let's not mess around with precession. That we need the spins to be doing their thing on their own, so we're just going to not bother with fractionalizing that. That's a bridge too far. The net result is you can still do MR imaging. You can still make really nice pictures of the brain, image-wise. The resolution here, sub-millimeter. This is a human brain in coronal section. And the map here of the diffusion coefficient shows you light or dark blue areas that are related to the values of the diffusion coefficient. So the higher diffusion coefficients are here in the light blue, and the lower diffusion coefficients there in the blue. With the fractional model, as I showed you earlier, you fit the same data, and the convergence gives you the alpha and beta values. In this case, the alpha and alpha, beta, mu, and tau values. And so you end up getting parameters that allow you to characterize the white matter, gray matter, CSF compartments in this slice of the brain with different factors that may or may not reflect different aspects of the microstructure. And that's, as I said earlier, is where the important questions can be posed, because it's the change in microstructure that happens with age, with Alzheimer's, with Parkinson's, with any kind of neurodegenerative disease that we would like an early view of. And you can't see it at pathology until you take a brain sample, which is not possible, and you can't see it in a conventional MRI. So the first representation of this is it actually leads to a stretched exponential, and this was good. I did this decade ago because the stretched exponential model had been fit, but here we showed that the fractional order wasn't just an ad hoc parameter; it could be viewed as the order of the space derivative. And if you think in general of how you could modify things, you could envision the diffusion equation as being diffusion in time of order alpha and diffusion in space of order beta. And the solution of this, at least in the K-space, if you take a Fourier transform of this in space, is just the Mittag-Leffler. And it turns out that this model is almost exactly what you do in MRI. The wonderful thing about MRI is you don't collect data as a function of time and frequency and then take a Fourier transform or a Laplace transform or do all kinds of funny modeling with it. You collect the data line by line, and you collect that data in K-space. It's just like X-ray crystallography. You're collecting spatial frequencies, and you're looking at the decay of each spatial frequency. And when you make it diffusion weighting, this becomes a Q parameter and this becomes the diffusion time. So that in collecting diffusion weighted data, you are collecting the parameters of your diffusion model as it's projected to be varying with fractional time and space. And of course, we understand the Gaussian model occurs when beta, the space derivative, is 2, and alpha, the time derivative, is 1, right here. And then there's a linear relationship here back to the origin, and this corresponds to a mean squared displacement that looks like 2 alpha over beta. Anything that diffuses faster than that we call superdiffusion. Anything that goes slower than that we call subdiffusion. Anytime we get above and beyond out here in this range, it turns out that the processes will immediately collapse to Gaussian. So up here in this top section, you have more or less normal diffusion. So this model has been fit extensively to this data I showed you earlier. I showed you earlier data from the brain corresponding to CSF, gray matter, and white matter. And this is a rat brain, actually. And these little circles here are showing you that the signals that are acquired here are collected in different parts of the brain. And then we've fit the brain, all the tissue, to a generalized Mittag-Leffler model. And then this representation allows us to not just to make graphs and fit curves, but to fit every pixel in the image. And this is what, pays radiologists and doctors big bucks, because you get really good pictures of every pixel for every slice in the brain. And so here we get the diffusion map that we get before, but you also get maps of the alpha and beta decay rates. And you have to, since you've messed with time and space, you got to be careful about the units. So there are time and space parameters that or scale factors that have to be inserted. And if you want to combine time and space, you can mix everything together. And one of my students, Carson Ingo, developed a whole normalized spectral entropy, which sort of takes these fuzzy diagrams and makes a very pretty map. Again, if you're going to take it to the clinic, you don't want to think, oh, that fuzzy area might be a problem, Richard. No, you want to know, is that fuzzy area really smoothly transitioning between normal and diseased case, or better or worse with treatment? Okay, so good tissue contrast and resolution are important. For the animal studies, you can image for hours, for fixed tissues, half a day even. And so you can make really nice pictures. But it turns out to get clinical data, you've got to do everything in about 5 or 10 minutes. And so Tom Barrack and colleagues in the UK have developed some schemes to acquire this data quickly from humans. And they're again fitting a Mittag-Leffler function. Again, the Q is the diffusion weighting term. That's a concatenation of the factors that go into B. And the delta is basically the diffusion time. And these models are fit in this case for a normal healthy subject, and it's and the data is collected in 3 or 4 minutes. Also, this model has been applied to a few patients. It's being examined now in clinical trials, but initially it's been looked at when you look at a brain tumor. With a relaxational agent, you can basically light the tumor up because of permeability in the blood-brain barrier. And you see differences in the pictures of the brain with normal diffusion and with the fractional order models. And it gives you information of perhaps regions around the tumor where the tumor or the normal tissue may or may not have been compromised. Other kinds of conditions have different patterns. And the goal of this sort of work is to examine the pathology. You're not looking for a tumor. Anyone who has a tumor like this has long been or much earlier been diagnosed. The question is, can we detect these sorts of things very early? And can we detect changes in the tissue with successful radiation or drug therapy very early, so that we know it's working? So these models are all based upon the continuous time random walk theory. There's another model of fractional calculus, as you know, that involves the using fractional motion. The CTRW model is nice because it can be sort of reorganized into something that looks like a modified Mittag-Leffler. So instead of having an E sub alpha x to the alpha, we've got an alpha and a beta here. Again, the data is collected as B, but we can make maps of different tissue structure. This is work that was done by Muge Carmen here in Chicago and with colleagues in China. The fractional motion model has some statistical insight in terms of it follows an exponential decay, but it assumes that the rate processes are time dependent. The statistics of this are reasonable, goes all the way back to Mandelbrot, but the model starts getting squirrely in that you get terms involving the fractional parameters, phi and psi here. They become additive and multiplicative. And this becomes degenerate in the sense that if you're trying to model a particular phenomena, you can get the same exponent here because it always appears as a sum or a product. And this leads to uncertainty in diagnosis, and it is a sensitive way of distinguishing, in this case, low grade and high grade tumors. So these are human brain tumors, gliomas in young children. Actually, the brains look small, as you can see, and it's very important to classify the type and to be able to quickly assess the response or non-response of the tumor to treatment. So we started with this picture. How do we inform the images? By using a model that employs fractional calculus. And I'll push this further. How do we choose the best model? If they're degenerate and if there's a whole Avogadro's number of models, then it's not clear which one is going to be best or better. And so what I've been doing recently, this is getting into the conjecture area, is trying to classify the models typically in terms of where they lie in a fractional cube or a space where you have an alpha and a beta here on the purple or I guess the orange plane here. Here's the time derivative, here's the space derivative, and then the vertical axis here is another parameter that we can change. Here, the diffusion constant is assumed to not change in time. But in the fractional motion model, you can interpret it as having a time-varying diffusion constant. For the case where beta equals 1, that is an exponential decay, or in the case where alpha is equal to 2, a Laplacian type motion here, then you get a similar face. So there are different faces of this cube that correspond to different slices of the set of models that you could build. One of your colleagues, Rubens, Irvin Lindsay, and I wrote a paper on this recently, actually. Just to show you a couple of tables here, and then I'll quit talking and we'll do some questions and answering. If you look at the alpha and beta models here for different parameters, here's the Gaussian model down here in the corner. Beta's 1, alpha's 2. And the conventional Levy walk, where you have a spatial derivative, is here. The subordinated Brownian motion here, where you have a fractional time derivative. And then you have the TTRW model that I've showed a couple of examples here, where we have fractional time, fractional space models. And this all basically takes place within this plane, regions of super diffusion. The mean squared displacement of the water goes as the diffusion coefficient times the diffusion time raised to a power that looks like beta over alpha. The fractional motion model, you have another set of the same kind of parameters. You get almost a degeneracy in terms of the mean squared displacement, because down here in the corner, the mean squared displacement goes as a 2 to the th, where the h now is this psi over phi parameter in the fractional motion model. And finally, for the fractional power law model, you get a diffusion mean squared displacement that goes as a nu plus beta term. So now we have these different models that have different terms that vary either the order of the time or space derivative or the space or the time dependence of the diffusion. Irving and some of his colleagues have done a lot of stuff with even space varying diffusion coefficients. The take-home message here is that the process can become degenerate again. When you start seeing things like nu plus beta, if that's going to be 3/4, you don't know whether that's 1/4 plus a half or a half plus 1/4, and it makes a big difference. So these models then are just viewed here as a way of classifying the mean squared displacements corresponding to the different fractional forms. That the conventional fractional model is powerful in that it generalizes ordinary diffusion, but it's just one of a set of time and space varying diffusion coefficients, which are linear differential equations or partial differential equations, but involve the actual integer order time. And here, fractional space, fractional time, integer order space. So it gets complicated. And you can choose whichever one you might use. The take-home message, I think, is that we're going to have to choose something more than time. And if you noticed earlier, I pointed out that the diffusion sensitizing factor, the B, is a product of the diffusion pulses and the diffusion time. So instead of just collecting data as a function of time or direction, we're going to have to collect it as a function of diffusion weighting. And that probably means a completely different type of data acquisition. Okay, I think I'm running out of time. In conclusion, I'll just try to point out that in general, in biological tissues, diffusion is anomalous. It is the new normal, as they keep saying nowadays. Anomalous diffusion is everywhere. It's in viscoelasticity. It's in dielectrics. It's almost anywhere you look, you find its fingerprint in the data. Fractional calculus models, we're biased. We're here at the conference. We like them, but they're versatile. That is, they're sort of like adjustable wrenches. You can adjust the order, and that's very pleasing because the mathematics, although challenging, the mathematics follows it interpolates between known values of order and known classes of models. Okay, I should acknowledge a lot of this data was collected by former students, Carson and Guanyon and Muge, and others with colleagues like Matt and Tom and Juan and Zhou Zhao. And the ultimate utility of this is being established, in the clinic, in the MRI community. But it's certainly a useful and a powerful way to model the complexity of progressive disease. Also, I should mention there's a special issue of the journal Mathematics, and it's all open source. So if you just click on this URL down here, you can get sort of the latest papers that we've been working on this topic, and so you'll be up to date. So I'll just close with two things. There is an anomalous relaxation and diffusion study group. It's based in Brisbane. You're welcome to join that and follow the lectures. In general, what we're talking about, I started with a Venn diagram, I'll end with a Venn diagram, is that we're using modeling and mathematics to inform and improve the diagnosis and treatment in medicine. So I think I will jump out of my talk here, if that's okay, Ruben. You there? Oops, I don't hear you.
Just fine, Professor. Thank for your great presentation.
Okay, am I doing okay on time? I figured I was.
More than okay. 15
Okay,
minutes for the questions. I'll bring
okay, good. I wanted
Carla
to
to
leave some
help
time.
us. Well, I'll show some the comments people did.
Okay.
The first one here, Professor Marian. I could hear your talk every day. Very interesting. And the other one is from Edmundo, that was with me at Portugal last time we were together.
Yeah. Do I know him? I may know him. Do I know him?
Yes, he was with me at Portugal. Remember?
Oh yeah.
When we go to the Fado show.
Yeah. In Lisbon? No.
Yes.
Yeah.
And
I
now
miss traveling. This is just not good. I want to go places.
It would be nice
never
to have
mind.
you
Okay.
here.
Oh,
Listen
good.
to some samba, show you our wonders.
Yeah. I'd love to meet your students and talk about fractional calculus.
It would be nice. Carla?
Okay, let's start with the questions. Vinicius asked how the fractional calculus could be applied to phase encode MRI, particularly phase contrast and Fourier velocity encoding.
Yeah, good question. I have not done that. I think that's a very good idea, and I think you should go do it. The phase encoding. The only trouble with phase encoding is you only get 2 pi. Once it goes around, it comes around. when you sample your as a function of angle, you don't get as many data points. But phase is the standard for elastography, for flow. The phase is used in many phenomena, and considering and I just go back to the MR equations, just brute force substitute where there's a time derivative, a fractional time derivative, and you know what you're going to get, and then look at that model as I was showing and fit the model against the data. Yeah, flow, even turbulent flow, I don't know. Flow through a complicated network or meshwork of a porous material. What we've seen with relaxation is that if you take and measure like the diffusion in a fluid, you get reasonable numbers. If you then put a bunch of little beads, plastic beads in that material.
Professor, we are having a problem with your microphone.
Oh no.
Okay.
Or maybe I knocked it aside. So if you're doing diffusion in a porous material, the diffusion coefficient goes down. But as long as you have sort of an aggregate of plastic spheres or glass spheres in your sample, the diffusion coefficient still will be Gaussian. It will just be reduced in magnitude. But if you put in small, medium, and large size plastic spheres, so that the space of the diffusion is different for different parts of it, then you start seeing this anomalous kind of multi-exponential behavior. And that's, of course, what you see in most complicated tissues. So, yeah, I have not done any work with fractional phase encoding. Be happy to talk to you about it sometime, and be happy to see what you come up with.
Very nice, Professor. Next one.
Next one was Felix who sent it. Is it correct to associate anomalous diffusion directly with Levy flights or Levy distribution?
If you read my book, you'll hardly see the word Levy flight or Levy distribution because it's only 694 pages long, and I admit as I wrote and thought about it, I did not understand enough about levy flights and levy distributions to even think about writing about it. It's only recently that I've become brave enough to even think about these things. The mathematics becomes very challenging with the spatial derivatives, and the super diffusion that arises becomes problematical in a physical sense, because although mathematically you can talk about in a CTRW model the particles being here and then all of a sudden they're being there, well, that involves they just moved at warp speed, no pun intended, to get there instantaneously. Now you can go back and adjust the model to account for a mean, a finite velocity, but the jumps that have to occur to get a levy process typically are associated with a turbulence, a flow, or some kind of a phenomena in addition to diffusion. It's not a bulk flow. It's not laminar, but there's something that causes the spins to leap. And it's very likely that this is what's going on. I mean, when you think about a tissue in the body with the blood vessels going through it, each blood vessel experiences a pulse and pressure with the contraction of the heart, and that pulse and pressure squirts a little blood. And if you look at the capillaries, the actual red blood cell, it's a little disc like this. It's too big to get through the capillary, so it folds over. And then it folds over, and then it pops open and squirts on. And so there's certainly some kind of super diffusion going on in there at all levels. And so it's reasonable to think that these models could portray some of that, but you're going to have to find a way to average over the cubic millimeters, which involve millions to billions of cells or blood vessels, thousands of blood vessels, to try to characterize that. But I think it's still, even in physical systems where you can control the onset of turbulence, the models are much more difficult to solve in the mathematical sense, and they're also difficult to interpret.
Very nice, Professor. Very interesting also. The next one is from a very good student.
I think you could do this one.
No, I would like to know a little bit about how did the academic community in the area receive your results? Did they know about the fractional calculus earlier?
No, they basically rejected. I mean, what do you do when you're confronted with something new and different? I mean, you attack it, right? I mean, wasn't it Gandhi that said, first they ignore you, then they attack you, then you win, right? Well, yeah.
No, but then, when they saw the results, were they impressive?
They are accepting of the results, but not of the interpretation.
Yeah.
And the problem, as I pointed out on those last slides, you have a degeneracy. You can envision a distribution of compartments, each of a different size, that could be characterized by a diffusion coefficient that varied in time and space as inverse power laws, with integer space and time derivatives. The mean squared displacements, as Irvin and others have shown, come out exactly as t to the alphas, t to the betas. And those alphas and betas can be stuck on the fractional derivatives in space and time, or they can be stuck on the diffusion coefficient. And which one do you want to use? I think there's a lot to be said for interpreting processes with memory and with non-locality. It just seems. I mean, look at van der Waals equation. I mean, in ideal gases, you start off with a single gas where there's no interaction with the other molecules, and then you admit, okay, it's got some physical size, so it has to have some repulsive, and it's got some attractive force because we know that things are condense. So you start looking at that, and there are factors that you have to be able to interpret. And I think the fractional models have the right statistical interpretation for modeling complex systems. And it works in dielectrics. It works in viscoelastic materials. It works in fluid mechanics. I mean, it works for almost everywhere we see it. But there's always a variety of ways of doing it. One can look at pseudo-differential operators. One can look at interpretations with nonlinear models. There are a lot of other ways you can add complexity to your system, and you have to sort of pick which one you want to use. Nice. But yeah, we have published it. There are a lot of groups doing it. I think it's going to be increasingly useful. But my first paper was rejected unanimously. The one I showed you that was published. I mean, I wrote probably the best letter I've ever written to the editor. I said, dear Professor whoever, you don't want to read this letter. I mean, you've already had my paper. You had 3 reviewers. They rejected it. It was clearly, I said, I've been an editor. I've written 100 papers. I agree with you 99 out of 100 times. But every once in a while, when something is summarily rejected, it's not wrong. There's a possibility that it might be useful or it might be different. And so the editor liked my letter, and so he sent it to 3 more reviewers. And he said, now all you have to do is get all 6 to agree.
Wow.
Very hard to work.
Yeah. So I mean, it's the way it works. But
next one
from Professor Carla. Do you have a parameter space, alpha or beta, which may characterize a specific brain disease? Biologically, what is the meaning of alpha or beta, if any? Thank you. Always a pleasure to listen to you.
Yeah, that's what we're looking for. And we've been looking at well-established animal models of degenerative brain disease. And so these are animals that have a degenerative brain condition that you can program in genetically, and then you can see it progress from week to week. And then you can look to see how the alphas and betas change. And what we're seeing is that in general, the alphas seem to be the most sensitive. That is this time-dependent factor. And that in many cases, we can set the beta equal to 2 and just leave it alone. But where I could be wrong is the question earlier about flow. It might just flip if you start applying fractional models to flow. It might very well work out that the betas become very important. But when you think about I worked for postdoc at the National Cancer Institute. I looked at a lot of mouse tumors. I looked at a lot of pathology. I talked to a lot of doctors, and the process by which a group of cells decide that they can no longer exist as a normal part of your body, and they start to mutate and then metastasize and develop their own little independent scheme, has different events occurring at different scales and different times. And if we can get a handle on those time scales and events and fit the alphas and betas to it, being that it's a complex structure, we might be able to establish early fingerprints for the likelihood of the disease progressing or not progressing. So I think it goes back to the physical interpretation, which is, what do you mean by the alpha? What do you mean by the beta? And I think in the fractional community, I use the CTRW model. The fractional motion model just says that there's some correlation in that if you jump to the right, the next time you're more likely to jump to the right. I mean, that's a reasonable thing to do. In kinetic theory, when you talk about collisions of gas molecules, I've been reading PChem textbooks, that there's something called persistence of velocities. That is, if you have some velocities in a collision, they aren't completely randomized. That a high velocity is more likely to continue in the direction that it's coming from. It's not the billiard ball collection. There's a persistent subvelocity, and that would show up in this fractional motion model, and that might be a way to connect a physical event with a mathematical term. But that's very important to establish. On one hand, the clinicians want, as I showed with the entropy, they want a clear-cut, pretty picture that's sensitive and specific. I mean, that's what I want. I mean, my brain's going to stop functioning someday. But if it starts, I want to know early, and I want to know exactly, what the problem is. With many conditions, you don't know till it's too late.
Very nice, Professor. That especially from a mother Alzheimer's disease, it would be a great step.
Yes. There is a lot of work being done on that.
Next one.
Next one's from Roberto. It's idea for his PhD thesis, he's saying. The question is, do you know something about fractional calculus applied to EEG forward problem?
That was easy. No. I mean, the trouble with fractional calculus, it's like the hammer theory. If you have a big enough hammer, everything looks like a nail. Fractional calculus is not the answer to every problem in the world. We can't have fractional calculus models of the universe, fractional quantum mechanics, fractional Schrodinger equations, fractional relativity. I mean, just because it's math doesn't mean that it's useful and needed. I come at it from the perspective that when there's a complexity in the interaction or in the associations of the conglomerate particles, then you're going to need fractional calculus. So I'll turn the question around in terms of the EEG. What aspects of, I mean, already that's a coordinated motion. It isn't just 10,000 axons firing at random. That's noise. Right, 10,000 axons firing randomly, it's just going to be white noise. You're not going to get 8 hertz, 12 hertz. You're not going to get you're not going to get evoked potentials. You're not going to get the periodic brain signals that are recorded. So there's already some coherence going on there, and I don't know of anybody who has looked into that aspect using fractional calculus. But if the data are not effectively modeled by what you're doing, it might be an opportunity to use fractional calculus. But then, as I said earlier, just because you can do it doesn't mean everyone is going to say it's a great idea.
Very nice. Professor, we are running out of time, but we have time for just to show that Manuel Ortigueira was here earlier. And he said it's always a pleasure to see you talking about the application of fractional calculus.
Yeah, I spent I guess a month a couple of times visiting in Lisbon with Manuel Ortigueira, and he's a very, very, very valued and interesting colleague. And I just thing I hate about COVID now is I just can't up and plan. I'm supposed to be at Cambridge this month. There's a big fractional calculus meeting going on at Cambridge, and I saw Igor Podlubny. He's there right now, and Blas van Agra is going next month. But I'm not going. I mean, I don't want to die. I mean, give me a break.
Please don't.
No,
Professor. Again, thank you very much for your great presentation. Hope to have you here in Brazil in the 3rd edition.
Oh, great. I look forward to meeting you again and visiting. Take care. Nice to meet you, Carla.
Nice to meet you too. Bye-bye.
Okay.
Pessoal, então encerramos aqui o dia de hoje. Encontro vocês amanhã. See you all tomorrow.
Okay. Bye-bye.
Bye-bye.
Bye.